Welcome to the video for species and speciation, where we will discuss the various methods scientists use to classify different species and the important role of reproductive isolation in the formation of new species, a process called speciation. Species are the fundamental evolutionary unit, which means that species evolve, individuals do not. Since species are what is influenced by evolution, it is important to be able to identify what a species is, but that can be quite complex. The most commonly used definition of a species is the biological species concept, which was proposed by Alfred Russel Wallace in 1865. The modern biological species concept defines species as groups of actually or potentially interbreeding populations that are reproductively isolated from other groups. Two individuals are reproductively isolated if they cannot have offspring or if their offspring are infertile. Thus, their genetics cannot be continued to be passed on. An example of two different species having infertile offspring occurs when a female horse mates with a male donkey. Their offspring is a mule, but mules are infertile and cannot have their own offspring. Thus, horses and donkeys are different species. It is also important to note the terms actually or potentially interbreeding populations. This means that two individuals are considered the same species if they can successfully breed, even if they do not do so in nature. For example, elephants living in India and Sri Lanka are considered the same species because if they are housed together in a zoo, they could have fertile offspring, even though they will never have offspring in nature because they cannot come into contact with one another due to their location. These two are considered subspecies, however, due to their geographic isolation, which we will discuss at the end of this video. While the biological species concept is the most commonly used definition of a species, it does have limitations. You must be able to determine if two individuals have reproductive success, which might be difficult to observe in nature. Additionally, not all organisms use sexual reproduction. Any organism that replicates asexually, such as bacteria, must be identified as a species in a different way. This is also true for organisms found in the fossil record. If you are trying to determine if two organisms that are now extinct were separate species, the biological species concept is not possible. Additionally, the biological species concept can be complicated by ring species. Two species who cannot breed, such as the two birds represented by the red and blue areas on this map. However, both birds do replicate with birds in the purple area. Thus, there is still a chance of gene flow between the populations, even though they are reproductively isolated. Biologists often use a second definition of species called the morphospecies concept, which states that members of the same species usually look alike. In this concept, if organisms have similar phenotypic characteristics, then they can be classified as the same species. This concept is useful, but it isn't perfect. For example, the variety of phenotypes we see in dog breeds does not make them different species. They are all members of the species Canis familiaris. While some species, such as the chimpanzee on the left and the bonobo on the right, look very similar, but are actually different species. Additional species concepts include the ecological species concept, which suggests that a species can sometimes be characterized by its ecological niche, which is the role the species plays in its environment. This includes habitat requirements and water and nutritional needs. The logic behind this species concept is that two different species cannot occupy the same niche due to competition over resources. For example, these two ladybugs are different species because they feed and mate on different types of plants. The phylogenetic species concept is based on the idea that members of a species all share a common ancestry, so those that cluster together based on phylogeny, or a diagrammed history of evolutionary relationships, are members of the same species. This concept is utilized more often now that we can look at the DNA sequence of organisms and is commonly used method in prokaryotic biology. While there are other species concepts, the biological species concept is still the most commonly used definition. remember that a condition of the biological species concept is that populations must be reproductively isolated in order to be considered separate species. Populations can be reproductively isolated, in other words, not able to have fertile offspring, for reasons that are classified as either prezygotic or postzygotic. Prezygotic barriers will be factors that prevent fertilization from occurring. Examples include habitat or ecological isolation, such as the water snake and terrestrial snake seen here. Because these snakes live in different habitats, they do not come into contact with each other to mate. Similarly, temporal isolation means that two species are looking to mate at different times, either different times of day or different times of the year, so their mating does not correspond. Behavioral isolation separates species because the mating behavior required to attract a mate is different between the species. In the picture here, the blue-footed booby, who lives on the Galapagos Islands, is performing a mating dance. The mating dance is meant to attract a female, and since the dance would not attract a female of a different species, these keeps the species separated. Alternatively, there are barriers that do not prevent an attempt at mating, but prevent the mating itself. These are mechanical isolation, where the physical act of mating is not possible because of the anatomy of the genitalia, or gametic isolation, where the egg and sperm are incompatible, as is the case with different species of sea anemones. There are some distinct species where members of the species can successfully mate, but postzygotic factors result in failure of the egg to develop into a fertile individual. These include reduced hybrid viability, where the offspring either does not come to term, or if it's born, it is born where it is unhealthy. Reduced hybrid fertility, which we discussed when we mentioned the horse and the donkey generating a mule. Other examples of this are a lion and a tiger mating to generate a liger. The third postzygotic method is hybrid breakdown, where the offspring of two different species may be able to mate, but the F2 generation is not. This occurs more often in plants than we'd see in animals. Populations evolve to become reproductively isolated from each other and thus evolve into different species. This occurs because once they are separated from each other, random mutations which occur over time appear in each population, causing the two to genetically diverge. Eventually, the two populations will have enough genetic differences that they are unable to interbreed. The easiest way to ensure genetic isolation is by physically separating the populations, so most models of speciation often focus on geography. When new species arise due to physical separation, it is called allopatric speciation. Because genetic divergence is typically gradual, we often find allopatric populations, populations that are separated by geography, that have yet to evolve reproductive isolation, but which have accumulated a few population-specific traits. This genetic distinctness is sometimes recognized by taxonomists, who call each geographic form a subspecies, as was mentioned earlier with the Indian and the Sri Lankan elephants. The geographical change required for allopatric speciation can occur by dispersal, where individuals colonize a distant place, such as an island, or by vicariance, where a geographic barrier arises within a population, such as a river that is rerouted or a change in landscape due to an earthquake. One of the best examples of vicariance-mediated speciation is 17 species of snapping shrimp that are distributed on both sides of the Isthmus of Panama. The Isthmus of Panama between Central and South America formed approximately 3.5 million years ago, separating the populations of marine organisms that could previously swim through the canal corridor between the Caribbean and the Pacific populations. Examining the evolutionary relatedness, or phylogeny, of these shrimp shows species with a distinctly paired pattern of relatedness. The closest relative of each species is one from the other side of the isthmus. This is consistent with the hypothesis that each of the Pacific-Caribbean pairings is derived from a single ancestor species indicated by a yellow dot in this phylogenetic tree. The continuous distribution between the Caribbean and the Eastern Pacific was disrupted by the formation of the isthmus, resulting in development of different species of shrimp over time. A good example of dispersal-derived speciation can be found in Darwin's finches. About 4 to 5 million years ago, volcanic action led to the formation of the Galapagos Islands, and eventually a small South American finch species arrived on those islands. The conditions on the islands were quite different from those on the South American continent, and each island had different conditions compared to the other islands. Thus, the isolated island populations underwent natural selection based on their new island habitats to become distinct from both the mainland ancestor and each other. It is possible, but much more difficult, for populations to undergo speciation without physical separation. Populations that are in the same place, that are not geographically separated, are called sympatric. Thus, this would be sympatric speciation, and is only possible if the population is under very strong disruptive natural selection, needed to overcome the gene flow between the two groups in the population.